All right, we are picking up with part 2 of chapter 43 on the immune system. So last time we talked about the innate immune system, and now we're going to focus on some details for the adaptive immune system. So when we're thinking of the adaptive immune system, remember that we're talking about a more complex and more evolved type of immune system. It's a more slowly responding system and a system that is important in establishing memory for our immune system, which is very helpful throughout the life of the organism. So our adaptive response will rely upon two different types of cells that are described as lymphocytes. Remember, cyte means cell, and lympho, the lymphatic system. The more common name for this are white blood cells, or you might see this abbreviated WBC. So white blood cells, which are the more common term for lymphocytes. So lymphocytes can be divided into two types, T cells and B cells. T cells are called T cells because they mature or are developed in the thymus, which is a organ found above the heart. That's part of the immune system, actually part of the lymphatic system. The immune system is part of the lymphatic system. The other type of lymphocyte that we need to know are called B cells. And B cells are called that because they mature or are developed in bone marrow, which is found inside of our long bones. Long bone examples would be your arms and your legs. So typically bones that are longer in length than they are wide. So those would be long bones. So T cells and B cells are going to be the important types of cells that we're going to talk about. So some other general definitions that we need to know for our immune system response are antigens. So antigens are substances that can elicit a response from a B cell or a T cell. And antigens can be anything, really. Antigens could be pieces of food. They could be pieces of pollen. They could be pieces of dirt. In the case of things that could be pathogenic, they would be potentially things on the surface of bacterial cells or proteins on the surface of a virus. These are all things that could be described as antigens. So T or B cells are going to bind to antigens via molecules called antigen receptors that are going to bind to a very specific spot on one part of the pathogen. So again, a good example would be our SARS-CoV-2, which caused our most recent pandemic. The protein on the surface of the virus that we targeted the vaccine to was the spike protein. So that spike protein is the antigen itself. And then our immune system is able to recognize that spike protein via the antigen receptors on B cells and T cells. So in this case, they would be receptors that are specific to target just the spike protein of that SARS-CoV-2 virus. So antigens are the piece of the pathogen, and antigen receptors on our B and T cells are going to recognize them and allow for some sort of adaptive immune response to take place. So the one specific part of an antigen that is bound by that antigen receptor is called an epitope. So again, the antigen that I was talking about before, SARS-CoV-2, the spike protein. So there could be actually multiple epitopes, multiple small locations on that specific spike protein that could be recognized by different antigen receptors. So an epitope is just a piece of an antigen or one small section or part of an antigen that is recognized by an antigen receptor. So each individual B or T cell that will be developed in either the bone marrow or the thymus, they're specialized to recognize one specific type of molecule and, more importantly, one specific epitope on an antigen. So when we talk about how the B cells and the T cells are able to create these antigen receptors, we need to think about what is the generic structure of these receptors. So we're going to start with B cells first. So if we look back at this picture, you can see that the antigen receptors on a B cell look different than the receptors that are on the T cell. The B cell antigen receptors look like little Ys. They have a similar structure to antibodies, which will be released by B cells, whereas the mature T cells, their antigen receptor is more of a linear piece instead of the Y that we see on the B cells. So the generic structure of the antigen receptor on the B cell is this Y-shaped molecule that I just showed you a picture of. The Y-shaped molecule has two identical heavy chains and two identical light chains. So when we look at this up close, we can see that not only are there heavy and light chains, which are polypeptides or proteins, but there also are regions that are described as constant regions and variable regions. The constant regions vary or are not, the constant chains are similar in all of these specific antigen receptors on B cells, whereas the variable or the V regions are very different, right? So there are lots of differences in the variable regions. The constant regions, there are very few changes. So it is constant between all the different types of antigen receptors that we would find on B cells. The variable regions are important because this is what provides antigen specificity. So this is what allows for the memory cells in my body to SARS-CoV-2 to recognize that specific spike protein on that specific virus versus the variable regions on the antigen receptors for another molecule. So say a flu virus or chickenpox virus. So these are differences that we're going to see. So when we look at our B cell antigen receptors, remember that it's a Y-shaped molecule. It has two heavy chains. So you can see the heavy chains in the middle here, and two light chains. So they're called light chains because they are smaller. They are going to run faster on a gel, a protein gel that we would look at. So light chains are the smaller pieces, and then our heavy chains are in the middle. The heavy chains, you can see, have an area that is embedded into the plasma membrane of that B cell. So this heavy chain is what is going to anchor our antigen receptor into the surface of the B cell. Now you can also see that not only do we have two heavy chains and two light chains on my antigen receptor, but I have C regions, which are the constant regions. That's the regions all at the bottom here, okay? And the V region, which is the variable region of both my heavy and my light chains. The variable region is what is specific to that specific epitope or the region on that antigen. Okay, so variable regions are specific to only that one certain epitope on your antigen. The constant region, these are going to be the same as all other antigen receptors on the B cells. Okay, so important to remember that our B cell antigen receptor is going to be embedded into the membrane of our B cell. It looks like a Y. So the structure of this molecule looks like the letter Y. It's got two heavy chains, which are the larger of the polypeptide regions, and two light chains, which are attached to those heavy chains. And we have variable regions at the end, right? So on the arms of the Y here, this is what is going to bind to the epitope. And then we have constant regions, which are going to be the same between all these different types of antigen receptors. Okay, so what's going to happen is that when my antigen binds, or that specific epitope of my antigen binds to this antigen receptor, then B cell activation is going to begin. So this is a complex process. There's a lot of steps in this, but we're just going to learn the big picture ideas for this. What's going to happen after a B cell is activated is that cells will be created that secrete a soluble form of these Y proteins that are called antibodies. Antibodies also have a more official scientific name called immunoglobulin. So immunoglobulins are antibodies. IG for short is how we would abbreviate our antibodies. There's 5 different kinds of antibodies, and the type that's made depends on what is the cell location and also what is the function of that particular antibody. But in general, antibodies are called immunoglobulins and IG for short. Okay, so antibodies look very similar to our antigen receptors, but instead of our antibodies being bound on the surface as they are here in my B cell, a activated B cell will actually secrete and let go of antibodies that will circulate around in the blood supply or the lymphatic system and help with immune responses. So this is showing you an example of this process of B cell activation. So you can see here's a pathogen. So in this case, let's just say that this is a bacterial cell. This bacterial cell has antigens on the surface, and you can see that the antigens have not only one epitope, but multiple epitopes. So again, that's just one small section of an antigen that will be recognized by a specific antigen receptor on a B cell. So you can see that this antigen receptor will only bind to this epitope. It will not bind to this epitope on this antigen here. Now, once this happens, again, I have B cell activation. More B cells are created, and these B cells are going to release antibodies that are specific to this particular epitope as well. And this binding of these antibodies to this pathogen will cause the cells to be killed in different ways. And we'll talk about what some of those ways are. Down here, this is just showing you that our antigen receptor is very specific to that one type of epitope. So again, in this case, you saw that there were different types of epitopes on this antigen. So antibody A is specific to this pointy area of this antigen. Antibody B is specific to the rounded area. Antigen C is specific to something else, right? So not on this particular bacterial cell at all. So each antibody has one specific target, and that target is called an epitope on an antigen, which could be something that could become pathogenic. Okay, so that was B cells. So our T cells also have antigen receptors. Remember, we saw the picture of that. Let me go back here real quick. Here. So our mature T cells also have antigen receptors. They are linear. They do not look like Ys like we see on our B cells. So this is a linear antigen receptor on our mature T cell, also still embedded into the phospholipid bilayer. So the T cell antigen receptor consists of 2 different polypeptide chains. There are alpha and beta chains. And just like we saw with our B cell antigen receptors, there are variable and constant regions. So remember that the constant regions are the same between different antigen receptors. It's the variable region at the ends or the tips of the T cell antigen receptor that are unique to each of those individual epitopes. Okay, so if we look at what this looks like, again, you can see a linear antigen receptor here. So you can see there are alpha chains, there are beta chains, and both the alpha and the beta chains have constant regions, which are embedded in the phospholipid bilayer of our T cell. You can see it also extends into the cytoplasm of the T cell as well. At the end or the tips of these T cell antigen receptors are the variable regions, and the variable regions are specific to certain epitopes. So the T cells are going to bind to these antigen fragments that are displayed or presented on a host cell. We're going to talk about this. So what this means is that the T cells are going to be moving around inside of our lymphatic system or our blood supply, looking for cells that are going to present or display pieces of potential pathogens. Okay, so antigen fragments are going to be bound to the surface of our antigen presenting cells, and they are going to be bound to what are called MHC molecules. And MHC molecules stand for major histocompatibility complex molecules. We're going to see that MHC molecules can be of type 1 or type 2, depending on the type of cell that you're talking about. But the main thing that's going to happen is that these MHC molecules are going to help T cells get activated by displaying pieces or parts of potential pathogens. So they're displaying antigens on the surface of the cells that allow for the immune system to know that there could be a potential problem afoot. So when a cell is infected, so say you are infected with some sort of bacteria or a virus, the cell will be taken over, right, by that virus, taken over by that bacterial cell. And what's going to happen is that cell is going to recognize that it's been infected, that it's been invaded, and MHC molecules will be generated. They're going to bind to antigen fragments that are processed inside of the cell, and then they're going to display the MHC molecule bound to the antigen fragment on the surface of the cell. This is called antigen presentation. So it's kind of like picture a cell being taken over by a bacterial cell. Inside of the cell, the bacteria is broken down by specific organelles. So it could be broken down by lysosome, proteasomes, breaks that bacterial cell into pieces. The MHC molecule is generated inside of that infected cell, and then a piece of the bacterial cell is waved on the outside of the cell to alert the rest of the immune system that something is wrong. So it's kind of like waving a flag saying, hey, I've been invaded, I need help over here, right? So you might have think about like traditional pirates, right? Pirates, if they take over another ship, they would raise the pirate flag on that ship so that everybody knows that this is a pirate ship. Or if there was a castle that had been invaded and had been taken over by rebels, maybe they would wave a particular color flag. Maybe if that castle and its people decide that they want to give up, they don't want to fight, they're going to surrender, they raise the white flag, right? So these antigen presenting molecules, the MHC molecules, which are presenting the antigens, are giving information to the rest of the cells that allow for T cell response to take place. So here's an example of what this would look like. So again, here is a pathogen. So say this is a bacterial cell. It is going to invade this host cell. The host cell is going to break down this bacterial cell into little pieces and parts. The MHC molecule is generated by the host cell, and this antigen is attached to the MHC molecule. It's moved out to the outside of the cell and it's displayed on the outside of the cell. So this is a cell that is saying to the rest of the cells around it and to our immune system, hey, I've been invaded, I've been infected, here is what has infected me. Then you have T cells floating around, and T cells will come and see that there's an MHC molecule that has displayed a foreign pathogen, an antigen, and it's going to bind to that receptor. That will allow for T cells to propagate, to make new T cells, and to make lots more T cells. So it's this antigen presentation process by these, what we describe as antigen presenting cells, that allow for T cells to be activated. And if, again, looking up closer at how this works, you can see that there's the MHC molecule sandwiched around the antigen fragment. So that T cell is going to be able to bind and recognize that there is an infection inside of this cell. So when we talk about B cell and T cell development, 4 major characteristics of this process. And again, we're just looking at an overview of this. There's a lot more details on how all of this works. but 4 major characteristics of B cell and T cell development in the adaptive immune system. So huge diversity in the lymphocytes and the receptors, and that all comes from the complex genetics of the variable regions of the B cell antigen receptor and the T cell antigen receptor as well. There is a property called self-tolerance. This has that the adaptive immune system will not react against an animal's own molecules and cells, so it is tolerant of molecules that are described as self molecules or self cells. So we want to make sure that it's only attacking things that are foreign, right? Foreign pathogens, foreign waste products, things that have come in from the outside. So self-tolerance, tolerating and not attacking self cells. B and T cells have to proliferate, meaning make more after they're activated. And a really big part of this is the fact that B and T cells have to be created to retain immunological memory, meaning that there are memory cells, memory B cells and memory T cells that float around in your blood supply that will recognize an antigen that has been seen before. And once this recognition happens, this is going to aid the immune system to respond more quickly, more efficiently, and sometimes will respond so quickly that the person doesn't even know that they were infected with a virus or a bacterial cell. That's the beauty, and we use immunological memory for our adaptive immune system, and we can use it artificially by creating vaccines. So we're going to talk about what vaccines do. Okay, so again, this idea of self-tolerance. So remember we said that our adaptive immune system has to be really good at telling the difference between self and non-self cells. So the antigen receptors, those B antigen cell receptors and T antigen cell receptors, they're generated by random rearrangements of DNA. Again, very complex genetic systems, which are super interesting to learn more about. As the lymphocytes mature in either the bone marrow or the thymus, remember B cells mature in the bone marrow, T cells mature in the thymus. Once they are maturing, or as they're maturing and creating new B and T cells, they are tested for self-reactivity. So if they test and are able to bind to self cells, then they are often targeted for apoptosis. Remember, apoptosis is programmed cell death. If they are binding to self cells and they're not targeted for apoptosis, they are actually going to be rendered non-functional. So we want to make sure that any newly created B or T cells that are positive for self cells or able to react to a self cell, that they are either killed or that they are created or rendered non-functional. We want to turn off that reactivity. All right. Well, we are going to pick up here in the next section of our chapter 43. All right. Talk to you soon. Bye.